Today we are going to discuss about the synthesis of steroid hormones. We all know that steroid hormones are being synthesized from cholesterol, and the major sites of synthesis is mainly gonads and adrenal cortex. Let's have some basic information about the steroid hormones, and later on we will learn the synthesis of these steroid hormones. We all know that steroid hormones are being synthesized from cholesterol. Cholesterol is nothing but a 27 carbon molecule. If we see this structure of cholesterol, it has got 27 carbon. This is the first carbon, second carbon, third and fourth. In the third carbon there is a hydroxyl group attached to, and this is fourth, this is fifth, sixth. There is a double bond between fifth and sixth carbon. This is seventh, this is eighth carbon, this is ninth, and this is 10th. This is 11th, 12th, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, and 27. So there are certain things important here, that and on the third carbon there is a hydroxyl group attached, and on the between fifth and sixth carbon there is a double bond, or we can say this double bond starts at the carbon number 5. There is a side chain at carbon number 20. This is 18, 19. Along with that, there are 3 hexagonal rings and 1 pentagonal ring. All these rings are fused together to make the steroid nucleus. Either it is vitamin D or any steroid hormones, or as well as cholesterol, all contains these 4 fused ring structure. The different types of steroidal hormones in humans are progesterone, glucocorticoids, mineralocorticoids. These all 3 contains 21 carbon in their structure. It means that, and all of them are being synthesized from cholesterol. It means that 6 carbon is being cleaved out from the cholesterol to make these hormones like progesterone, glucocorticoids, and mineralocorticoids. And later on, it will be the 2 carbon will be further reduced to make the androgens, and 1 carbon will be cleaved out. Along with aromatization, estrogens will be synthesized. There are 3 major sources of cholesterol in our body. First of all, depending upon the importance, LDL cholesterol. The steroidogenic tissues take up the cholesterol by receptor-mediated uptake through LDL and use it inside the cell for steroid synthesis. The second important source is the de novo synthesis. The cholesterol is synthesized from acetyl coenzyme A by using cholesterol biosynthetic pathway, and we have I think we have learned this in another topic. And the third and least important is from the HDL through the scavenger receptor beta 1. Although in certain animals, like specifically rat, it is the major source. However, in humans, plasma LDL cholesterol is the major source. And we all know that some of these hormones are being synthesized at certain specific location in the body. However, all of these hormones can be synthesized in other places as well. Like approximately all hormones are being synthesized in adrenal cortex, but there are certain places or certain organs in our body that are producing these hormones in high quantity, or we can call it the major site for the synthesis of these hormones. So the progesterone is basically synthesized in the corpus luteum. This is the corpus luteum in the ovary, and the estrogens are mainly synthesized in the ovaries. This is the primary sex organ among the females, 2 ovaries. This is uterus and uterine tube and fallopian tube. And androgens, or the primary androgen is the testosterone, is being synthesized in the testis or in the Leydig cells. This is the structure of a testis. And lastly, but not the least, the glucocorticoids and mineralocorticoids are being synthesized in the adrenal gland. Adrenal gland is present over the kidney. If you see this, these are the adrenal gland. This is the kidney. These are the kidneys. And in the adrenal gland, there are two major structures. One is the cortex, and the other one is the central part that is known as medulla. Medulla is responsible for the synthesis of epinephrine and norepinephrine. However, other steroids are being synthesized in the cortex part. Cortex has been further divided into three major layers. The outermost layer is known as zona glomerulosa, and then in between layer is known as zona fasciculata, and the innermost layer that is adjacent to the medulla is known as zona reticularis. So we have to know these layers because zona glomerulosa mainly synthesizes the aldosterone or mineralocorticoids, and zona fasciculata is mainly responsible for the synthesis of glucocorticoids. Therefore, in future, in foregoing discussion, it is important to note that these three layers are present in the adrenal cortex, and all layers are responsible for the synthesis of different hormones. However, the primary enzymes are present in every layer, but the differentiating enzyme or the later enzyme that finally result for the synthesis of a particular hormone like cortisol or aldosterone is only present in certain layers. Now, we will a little bit discuss about the individual hormone. We will not go into the detail, but because these things will be covered in another like physiology and other discipline. But a little bit we have to see. Like for example, glucocorticoids is having 21 carbon in their structure, and in the human, the primary glucocorticoid is the cortisol that is being synthesized in the zona fasciculata. We have recently discussed of adrenal gland, and the major functions or actions of the glucocorticoids or cortisol is the regulation of gluconeogenesis, as we have learned this topic in introductory biochemistry in second year, and it also regulate or enhance the degradation of fats and proteins and inhibit immune and inflammatory response. They act as immunomodulator in our body, and this hormone is also responsible for the responsiveness towards the stress. It also elevates blood pressure and sodium uptake. However, this action is primarily done by aldosterone, but in the larger quantity, cortisol or glucocorticoids can also do the same job. This is the structure of cortisol. As we see here, normally in the cholesterol there is a double bond here between carbon number 5 and 6, but in the cortisol or any steroidal hormones, a majority of the steroidal hormones having the double bond is here in the between carbon number 4 and 5. Mineralocorticoids, the primary mineralocorticoid in our body is aldosterone. It is also a 21 carbon hormone, steroid hormone, but this is produced mainly in the zona glomerulosa, unlike the cortisol that is mainly produced in the zona fasciculata. And we know that zona fasciculata is the middle layer. However, the zona glomerulosa is the outermost layer of the adrenal cortex. And the primary function of aldosterone is to increase reabsorption of sodium and excretion of potassium and hydrogen, and that will lead to increase in the blood volume as well as blood pressure. So along with the renin-angiotensin aldosterone system, it is the primary regulator of blood pressure in our body. This is the structure of aldosterone. Now the progesterone. Progesterone is also having the 21 carbon in their structure, and it is mainly produced, as we have discussed, in the corpus luteum. responsible for the luteal phases of menstrual cycle, and it is also act as a differentiating factor for mammary gland among the females. It prepares the lining of endometrium for implantation of ovum, and progesterone is also essential for the maintenance of pregnancy. And lastly is the sex organs, sex hormones. This is the structure of the progesterone. Now we are coming to the sex hormone. This estrogen is the mainly female sex hormones. There are two major types of estrogens in our body or in a female body. In male also, because it is responsible for the fusion of epiphysis. Estrone and estradiol. These hormones are required for the development of secondary sexual characters among the females. Estrone along with progesterone also participate in ovarian cycle. This is the structure of estrogens. Now the last is the androgen, and primary androgen is the testosterone in our body. This is the male sex hormone, and it produces all the secondary sexual characteristic among the males. It is synthesized from progesterone by sequential pathways. We will discuss in foregoing slides. Now there are certain things required for the synthesis of steroid hormones, like for example anything that is being synthesized in our body needs enzymes, and there must be some location for the synthesis of those particular substances. So we will discuss about the different types of enzymes that participate in the synthesis of steroidal hormones in our body, and what is their intracellular location. There are two major different categories of enzymes that are responsible in the synthesis of steroidal hormones. One is the cytochrome P450. It is also known as monoamine oxidase, cytochrome oxidase P450. There are a lot of names, but for convenience and for simplification, I had written only one to avoid confusion. But in other books, there are multiple names, so you have to know other names as well to avoid the confusion. There are a variety of names, but they may be also known as only P450 enzymes. Everything is okay. And the other is the hydroxysteroid dehydrogenase, and popularly known as dehydrogenase only. There are multiple types of dehydrogenase. We will see in future slides what are different types that are responsible in the synthesis of steroid. Now the intracellular location is the first and committed step in the synthesis of steroid, takes place in the mitochondria. However, other places is all the sequential reaction takes place in other intracellular organelles like microsome, endoplasmic reticulum, and even in the cytoplasm. So these are the places where these hormones will get synthesized. Now there are two major enzymes. One is the cytochrome P450 and the other one is the dehydrogenases. In the cytochrome P450 or simply P450, there are two types of enzyme, type 1 and type 2. In humans, there are 57 different types of P450 enzymes, and type 1 approximately 7 different types of type 1. Type 1 exclusively present in the mitochondria and need adenodoxin and adenodoxin reductase as electron acceptors. The first enzyme that is responsible for the synthesis of steroids is P450 SCC or P450 side chain cleavage, also known as desmolase. For simplicity, we will use the word desmolase, that is more easier for us to remember. Is a type of type 1 P450 enzymes and need ferredoxin, also known as adenodoxin, and ferredoxin reductase, also known as adenodoxin reductase, for optimum activity, because these enzymes utilizes NADPH, takes up the electron from the NADPH and transfer it to the substrate and make the product. This is the usual scenario. These all 7 different types of enzymes are present in the mitochondria. And the next is the type 2. Type 2 is the microsomal enzyme mainly present in the endoplasmic reticulum, and it also needs another P450 enzyme for optimum activity, that is known as P450 oxidoreductase, or popular name is POR. Now the next is the hydroxysteroid dehydrogenases, or maybe only dehydrogenases. They have been classified depending upon the two basis, that is the structural basis and functional basis. And structurally they further divided into two different types: short chain dehydrogenase reductase, aldoketo reductase, and functionally there are multiple types: 3-beta hydroxysteroid dehydrogenase type 1 and type 2, 3-alpha hydroxysteroid dehydrogenases, 11-beta hydroxysteroid dehydrogenases, and 17-beta hydroxysteroid dehydrogenases type 1, type 2, type 3, type 4, and type 5. Only 3-alpha hydroxysteroid dehydrogenases does not take any participation in the synthesis of steroid hormones. The rest, all of them are being utilized in the synthesis of steroid hormones. Now the first and committed step in the synthesis of steroid hormones, or we also call as steroidogenesis, is the synthesis of pregnenolone from cholesterol. Pregnenolone is a 21 carbon molecule, and cholesterol is a 27 carbon molecule. So this step means the cleavage of 6 carbon, or taking out of 6 carbon from the cholesterol. However, this reaction takes place in 3 different steps, 3 different sub-steps. The first step is the 22 hydroxylation of cholesterol, and the second step is the 20 hydroxylation of 22 hydroxycholesterol, and then oxidative cleavage of 20-22 dihydroxycholesterol, and that leads to cleavage or taking out of the 6 carbon from the cholesterol. So now the remaining structure will become 21 carbon, and in this reaction 3 moles of NADPH as electron donor and oxygen is utilized, and the reaction is catalyzed by desmolase enzyme, and we all know that this enzyme require ferredoxin as well as ferredoxin reductase, or adenodoxin reductase for optimum activity. So this is the sequential reaction. Cholesterol is converted into pregnenolone, and this is 21 carbon molecule, this is 27 carbon molecule. 6 carbon will get out or cleaved out. If you see, approximately the structure is the same. There is no much difference, except for there is hydroxylation at this position number 20. This is the 21 carbon, this is the 20th or 20. Now the next step is the synthesis of other steroid hormones like progesterone, aldosterone, cortisol, or from pregnenolone. So progesterone will be converted into oxidized and isomerized to make pregnenolone will be oxidized and isomerized to make the progesterone. So here is the pregnenolone. If you see here, this is the double bond present in between carbon number 5 and 6. However, in the progesterone, this double bond changes the location. That is, isomerization takes place, and carbon number 4 between this here it is between carbon number 5 and 6 in pregnenolone. In case of progesterone, it will isomerize to carbon number 4 and 5. This is carbon number 4, this is carbon number 5. Moreover, this carbon number 3, as we see here, there is a hydroxyl group in pregnenolone. However, in case of progesterone, it has been oxidized. As we know that first is the oxidation and then isomerization. So there is oxidation here and then isomerization here. That will lead to the synthesis of progesterone, that is 21 carbon carbon number is same, there is no decarboxylation, and this enzyme 3-beta dehydrogenase is used or act as a enzyme in this reaction. Now next is the synthesis of aldosterone. In the aldosterone, the first of all, the progesterone is hydroxylated at 21 carbon number 21, followed by 3 sequential reaction: 11 beta hydroxylation, 18 carbon hydroxylation, and 18 carbon oxidation. So this will lead to finally lead to aldosterone, and these 3 reactions are catalyzed by a single enzyme P450C11 aldosterone synthase, or AS. However, in other books it is mentioned that 11 beta hydroxylase 1 and 2, but both these enzymatic activity are present in a single enzyme. So we have enumerated all 3 steps: 11 beta hydroxylation, 18 carbon hydroxylation, and 18 carbon oxidation. All 3 activities takes place by the same enzyme. So first of all, in the first reaction, the progesterone will be hydroxylated at 21 carbon to make the corticosterone, and then this reaction is catalyzed by 11 beta hydroxylase 1 and 2. Actually, these are the same enzyme as these enzymatic activity are present in aldosterone synthase enzyme. So the same enzyme has got multiple enzymatic activity, and finally aldosterone will be synthesized. If you see here the structure, here there is a hydroxylation, here there is a oxidation, and here there is a hydroxylation. Cortisol is also synthesized from the progesterone. We all know that 21 hydroxylation of pregnenolone will lead to progesterone. So after the synthesis of progesterone, there will be 3 sequential hydroxylation reactions: 17 alpha, 21 alpha, and 11 beta. Here it is 17 alpha, 21 alpha, and 11 beta. These 3 sequential reaction takes place at 17 carbon, 21 carbon, and 11 carbon by specific enzymes, that is 17 hydroxylase, 21 hydroxylase, and 11 beta hydroxylase. So this is 17 alpha hydroxylase, 21 alpha hydroxylase, and 11 beta hydroxylase. These 3 enzymes will finally lead to synthesis of cortisol. Progesterone will be hydroxylated at 17 position to make the 17 hydroxyprogesterone. This step does not take place in the synthesis of aldosterone, and then it is followed by the synthesis of 11 dehydroxycortisol by 21 alpha hydroxylase, followed by 11 beta hydroxylation that will synthesize the cortisol. Now the synthesis of androgens, or sex hormones, and estrogen from pregnenolone. Pregnenolone will lead to synthesis of progesterone, then androgen, then estrogens. We know that androgens are mainly responsible for male sexual characters, and estrogens are mainly responsible for female sexual characters. However, this is mainly present in male, but a little bit quantity is also present in female. This is mainly present in female, but a little bit quantity is required in male also. I think these things will be discussed in detail in another discipline. Why these hormones are present in different sex also. So androgen is major androgen in our body is the testosterone, and synthesis of androgen starts with the hydroxylation of progesterone at 17 location, like for the cortisol, as we have discussed right now. And the side chain consisting of C20 and C21 is then cleaved to yield androstenedione or androstenedione and androgen. Testosterone, another androgen, is formed by reduction of 17 keto group of androstenedione, and testosterone is reduced to yield dihydrotestosterone, a powerful androgen by the action of 5-alpha reductase 1 and 2. So in the first reaction we have already discussed, because this takes place in cortisol synthesis as well. In the next reaction there will be cleavage of one carbon, so that will form androstenedione, and then finally there will be reduction at the 17 keto group, and that will lead to synthesis of testosterone. Testosterone will lead to synthesis of dihydrotestosterone, that is more powerful, even more powerful than the testosterone, and by the enzyme 5-alpha reductase. Exactly in the same manner, the estrogen is synthesized by cleavage of one carbon and aromatization, that will lead to synthesis of estrogen. Estrogen is synthesized from androgens by loss of C19 angular methyl. We know that the 18 carbon as well as 19 carbon both are angular methyl group, and the formation of an aromatic A ring. Estrogen is derived from androstenedione, whereas estradiol, another estrogen, is formed from testosterone by the action of enzyme aromatase. Now we will look all these reaction in a single slide, how all these reaction takes place, and all these androgens and estrogens being synthesized from progesterone. So progesterone, first of all, there will be 17 alpha hydroxylation, that will lead to synthesis of 17 alpha hydroxyprogesterone, and then it will lead to synthesis of androstenedione, and then androstenedione will lead to after a cleavage of one carbon. So the in the first reaction there will be 17 alpha hydroxylation by the enzyme 17 hydroxylase. Progesterone will be converted into 17 alpha hydroxyprogesterone. We know that this reaction also takes place in the synthesis of cortisol, and then in the next reaction there will be a cleavage reaction, or the one of the carbon will be cleaved out from the 17 alpha hydroxyprogesterone, that will lead to androstenedione, and then in the later reaction there will be reduction reaction, alpha keto reduction, that is by the enzyme 17 keto reductase, and finally the testosterone will be synthesized. Later on in the subsequent reaction, either at the androstenedione or at the testosterone level, first of all we will discuss in the androstenedione, by the action of aromatase enzyme, estrone will be synthesized, and in the testosterone, estradiol will be synthesized by the enzyme, same enzyme, that is aromatase. Now this is the slide that will tell us how the testosterone and androstenedione will be converted into estradiol and estrone respectively by the same enzyme aromatase, and moreover the testosterone as well as androstenedione both can be converted into each other, or we can say that it is a bidirectional reaction, can occur in both the directions by the same enzyme, and exactly in the same manner estradiol and estrione can also be converted in one another molecule by the same enzyme 17 keto reductase. So now we are going to discuss about some abnormality due to defect in the enzymes of the synthesis of steroid hormones, and the most common of these abnormalities is the congenital adrenal hyperplasia. But this disease like congenital adrenal hyperplasia is one of the most common autosomal recessive disorder in born error of metabolism. However, we would like to have some revision whatever we have discussed so far. So we know that cholesterol, that is the 27 carbon molecule, there is a 6 carbon cleavage from this structure by the enzyme desmolase, and we know these multiple reactions. So there are 3 different reactions. First of all, there is a 20 hydroxylation, then 22 hydroxylation of 20 hydroxycholesterol, and then cleavage of 6 carbon will be cleaved out from the 22 dihydroxycholesterol. So finally pregnenolone will be formed, and this pregnenolone is 21 carbon. Then pregnenolone will lead to synthesis of progesterone. This progesterone will lead to synthesis of 17 alpha hydroxyprogesterone. However, in case of synthesis of aldosterone, because in the synthesis of aldosterone this step does not take place, so in case of aldosterone synthesis, this progesterone will be hydroxylated at 21 alpha carbon by the enzyme 21 alpha hydroxylase, and that lead to synthesis of 11 deoxycorticosterone, and then this will be further hydroxylated at 11 beta hydroxylase, and that will lead to synthesis of corticosterone, and then finally by the 11 aldosterone synthase or 11 beta hydroxylase to aldosterone. In the next reaction, the 17 in the synthesis of cortisol, 17 alpha hydroxy progesterone will be hydroxylated at 21 carbon by 21 alpha hydroxylase, and this will lead to the synthesis of 11 deoxycortisol, and further this 11 deoxycortisol will be hydroxylated at 11 position by 11 beta hydroxylase, and then there will be synthesis of cortisol. And the androgens as well as the estrogens will be synthesized from this 17 alpha hydroxyprogesterone. The first reaction, there will be a cleavage reaction. One carbon will be cleaved out. So two carbon will be cleaved out, So two carbon will be cleaved out to make androstenedione, and then this will be further a reduction or keto reduction will take place and converted into testosterone, and then testosterone, from the testosterone one carbon will be cleaved out, and aromatization will lead to synthesis of estradiol, and androstenedione may also lead to estrone by the action of the aromatase enzyme. So this is what we have discussed so far. But what happens when certain of the enzymes is being defective or deficient in this pathway? Like for example, in 3-beta dehydrogenase deficiency, this hormone 3-beta dehydrogenase is being deficient or defective. So what will happen? There will be increased concentration of pregnenolone, but there is a decreased concentration of progesterone. So no glucocorticoids, as we have seen here. If this enzyme is being blocked or defective or deficient, what will happen? This will not be synthesized. This will not be synthesized. Either of them are not being synthesized. So no glucocorticoids, no mineralocorticoids, no androgens or estrogens will be synthesized. Increased pregnenolone or 17 hydroxypregnenolone, since as we know that this pregnenolone is converted into progesterone by 3-beta dehydrogenase, but sometimes this pregnenolone can be converted into 17 hydroxypregnenolone by this enzyme 17 alpha hydroxylase. Since this enzyme is active, the majority of the pregnenolone can be converted into 17 alpha pregnenolone, and that will lead to the synthesis of DHEA and DHES. Now there will be a marked salt excretion because there is a decrease or absence of mineralocorticoids. Therefore sodium reabsorption either does not take place or take place at a very slow pace or a very suboptimal, and finally will lead to salt excretion in increased quantity. All patients have female genitalia because of the lack of androgens. In another type of deficiency or another deficiency of enzyme, that is the 17 alpha hydroxylase enzyme. In this case, this enzyme 17 alpha hydroxylase is being deficient, defective or abnormally function So what happened? This progesterone is being synthesized, and this progesterone can be hydroxylated into 11-deoxycorticosterone. So what will happen? This pathway is normally functioning, the synthesis of aldosterone. Therefore, all the and this pathway is completely or partially blocked, depending upon the severity of the disease. So what will happen? All of the cholesterol that is going through this pathway will make the aldosterone. So what will happen? Of course, the aldosterone is responsible for the increased absorption of sodium and increased excretion of potassium and hydrogen, and increased blood pressure as well as blood volume. So what will happen? And there won't be any estradiol or testosterone or maybe any cortisol. So what will happen? No sex hormone or cortisol are produced. Low level of 17-hydroxylated C19 and C21 steroids. Increased production of mineralocorticoids causes sodium and fluid retention and therefore hypertension. So we know that since the pathway responsible for the synthesis of mineralocorticoid is normal, and because of the blockage of the synthesis of cortisol as well as androgens and estrogens, what will happen? All the pregnenolone or progesterone will be diverted toward the 21 alpha hydroxylation and then the formation of aldosterone. So there will be a high quantity of aldosterone is produced, and more sodium will be reabsorbed, and high blood volume as well as high blood pressure will be seen among the patient. Affected male patients with pseudohermaphroditism, because pseudohermaphroditism is having the ambiguous genitalia. They are having genitals more look like female. So just because of the deficiency of the sex hormones in the male, and among the females, they are unable to reach the puberty because the lack of estrogens, and they have the infantile genitalia because the secondary sexual characters need estrogen synthesis, and we know that there is a decreased or absent synthesis of estrogen, female sex hormone. Lastly but not the least, because this is more important, and majority of the patients suffering from congenital adrenal hyperplasia is suffering from 21 alpha hydroxylase deficiency. This hormone synthesizes the 21 alpha hydroxylation and will lead to synthesis of 11-deoxycorticosterone or 11-deoxycortisol. Finally, the respectively desynthesize aldosterone and cortisol. So what will happen? There will be increased synthesis of androgen or estrogen, but there is a decreased synthesis of mineralocorticoids and glucocorticoids. So let's see what will happen. There may be three different presentations. The most severe of them is also known as salt wasting congenital adrenal hyperplasia, in which there will be very low blood pressure in the patient, and multiple cardiogenic shocks, and these episodes used to occur in those patients because the patient severely lacking the mineralocorticoid synthesis, and there is a very low level of mineralocorticoids or absent mineralocorticoids in their blood. In the moderate type, there will be simple virilizing congenital adrenal hyperplasia. In this case, what happens? Some of the mineralocorticoid and some of the glucocorticoid are being synthesized, but since the increased quantity of androgen are being synthesized, the virilization takes place in the female as well as in the younger males. Non-classical CAH, congenital adrenal hyperplasia. It is a mild type of CAH. This is the most common type of congenital adrenal hyperplasia, that is the deficiency or defect in the 21 alpha hydroxylase. And these deficiency or defect may result in three different presentations depending upon the severity of the disease. If it is all the enzyme is absent or very less than 10% is functional, this is severe. If some of the enzymes are still functional, it is known as moderate, and majority of the enzyme is functional, a little bit is defective, then it is mild. Mineralocorticoids and glucocorticoids are deficient, as we have discussed already. And overproduction of androgen lead to masculinization of external genitalia in females also, and early virilization in male. Increased potassium and decreased sodium in the plasma